All right. Good afternoon, everyone. Sorry, I know there are still people coming in, so please don't feel rushed to grab your knitting materials. Okay, so welcome to Knitting with Neuroscience, meeting the researchers. My name is Olivia Kelly, and I'm a cultural producer here at the City of Sydney. I'd like to start by acknowledging that the City of Sydney conducts its work on the traditional lands of the Gadigal people of the Eora Nation. I'd like to pay my respects to elders past and present and extend that respect to any First Nations attendees joining us today. So I just have a few housekeeping things before I hand over to the speakers. So we are recording today's talk through Zoom. You may have noticed the camera at the back there. If you're joining us on Zoom, the best way to view the talk is to choose the Fit to View window from the View Options drop down at the top of your screen. This talk is being interpreted in Auslan, and we thank our fantastic interpreters today. We will have a Q&A session at the end of the talk. So we'll be taking questions from within the room and also via Zoom. You can submit your questions via the Q&A or the chat feature on Zoom. We've created a short survey to find out about your experience today. So I'll put up a QR code at the end of the talk, and I'll also email out that survey link to everyone who signed up. And I'll also let you all know that we do have a photographer here today who's going to be taking some snaps. They'll just be kind of broader group snaps, so there won't be anyone individually, but if you feel uncomfortable being photographed, just let us know. Okay, so today we're joined by four fantastic researchers. Dr. Stela Petkova, Dr. Vivian Liao, Dr. Nathan Cross, and Dr. Anastasia Suryev. Our researchers will be talking about brain health across your lifespan, covering autism, anxiety, epilepsy, and sleep. We're also joined by Pat and Rita, who are the founders of Neural Knitworks, which started over 10 years ago as a mindfulness project. They're just at the sides of the room, either side, and they can give you some guidance on how to knit a neuron. But I can see a lot of people have already gotten well ahead, like the neurons are looking very, very beautiful. And the talk today is a part of National Science Week as well. So I'll now hand over to our first speaker, Dr. Stela Petkova. Thank you.
Hello, everyone. My name is Stella. I'm a researcher at the Brain and Mind Center, and my work centers on how we can use mice to learn more about autism and social anxiety. And so today I'm going to just give some really broad insights into how we think about autism and how we're using what are called preclinical models, so not people, but animals. And by the end, I hope you see why we use these small little guys to make big steps in understanding autism. So I pulled together some images when I Googled what is autism portrayed by media. And while this portrayal is evolving, going from Rain Man to some more modern portrayals, and it's getting more nuanced as we go, it's still straying quite a bit from what autism actually is. So I feel like our tiny step number one should be, what is autism? So autism is a neurodevelopmental disorder, meaning it originates in the brain and happens during development or begins during development. It's defined behaviorally, which means there's no blood test, there's no scan a doctor can do to diagnose it. It's all in our behavior. And while some kids get diagnosed earlier, it's typically diagnosed around 4 years of age or older. And it's quite prevalent. So currently in Australia, about 1 in 100 Australians will be diagnosed with autism, which means that about 250,000 of Australians are diagnosed, which also means likely every one of us in this room and online probably knows someone who is on the autism spectrum. And about 70% of these people have other conditions or other disorders that they live with, such as intellectual disability, physical disability, epilepsy or seizures, or anxiety, social or generalized. And so, again, like I said, autism is diagnosed behaviorally based on 2 very broad primary diagnostic criteria. So one is impairments in social communication interaction. This is what we think about when people can't maintain proper eye contact or are nonverbal. And the second is restricted or repetitive behaviors. This can include like rocking or stimming or even being really interested in trains. But like I said, It's truly a spectrum, and it's really complicated because the reality looks more like this. It's truly a spectrum with a lot of variation and a lot of other co-occurring conditions and considerations when we think about people's care. And I've studied a lot of these different aspects throughout my scientific career, and I'm currently focused on social behavior using mice and rats and understanding specific elements of social behavior. So tiny step 2, what causes autism? Autism, again, really complex. There are a lot of contributing factors to a child developing and being diagnosed as on the spectrum, but we can rule out a couple of things. So 1, bad parenting, or what used to be called refrigerator moms, do not cause autism. 2, vaccines, a long standing myth that has been debunked over and over again, also cannot cause autism. And anything related to a child's diet. This was a fad about a decade ago. You can feed your kids whatever. They're not, it's not going to impact whether they're on the spectrum or not. However, I could give a whole other hour-long talk on all the findings the field has made on the risk factors for developing autism. In the last few decades, we found many genes that are risk factors, environmental factors like pollutants or toxins, and even maternal influences like being really sick during pregnancy, or even some medications that are perfectly safe in other periods of our life, but that are not safe during pregnancy. It's really complicated, and this is where we can use mice and rats to start breaking it down. So mice and rats are great. And people can laugh at me when I say that, but they have truly revolutionized science and certainly the autism field. One way they've done that, which again, I could speak endlessly on, is understanding those causes of autism. All those elements I previously mentioned, we learned from studies with humans, but we're not able to actually determine which of those are causative to autism because humans are really complex. But we can break it down and model it simply in mice. We can manipulate mice in ways that we just can't with humans to better understand those risk factors in developing autism. It turns out we can test things like whether older parents is a risk factor for autism by using old mice. We can test whether medications pregnant moms take increase risk by injecting pregnant mice moms. And believe it or not, we can even determine genetic risk factors by looking at gene mutations in people and actually targeting those same genes in mice and seeing whether they have the same effects in humans. And then we could do all sorts of things like test their brains and their organs and their blood and even their behavior to see how closely it resembles autism like we see it in people. And behavior is my particular area of interest, and I study social behavior in mice. And I know I'm going to get this somewhere. Mice are not humans. Mice will never be children, but we're also not asking them to be. I'm going to quickly highlight 3 different kinds of social behavior that humans engage in and regularly, especially children, and how we actually can see those things in mice. So first example is social communication. Rodents will never talk. This is not Ratatouille, but they do produce vocalizations. We can't hear them, but they do make them, especially in the first few weeks of life. And so we can actually record these vocalizations because they're a way for the babies to get mom's attention and say, I'm cold, come get me, I want milk, I'm hungry, whatever. So we can do these experiments where we get mom and mom has babies, and we take each baby away from mom for a short amount of time, and babies cry out much like human babies cry out for their parents. And we can actually quantify to see how risk factors for autism actually change this. So the little graph, so the black dots are perfectly normal pups. They come from moms who had nothing done to them. And we see this peak of calls as they start to, as their larynx develops and they're starting to get more confident. And then as they get older, they develop fur, they open their eyes, they can move around. They don't need mom so much anymore, and they kind of stop calling for her. But our babies that were born from a mom with a risk factor, they actually don't produce as many calls. And this is actually very similar to a lot of first instances that parents report where their babies don't vocalize their needs, like I'm hungry, I'm tired, or wet. They just don't vocalize. So they're really, really, it's very different, but it's getting to the same point. Second is mice and rats play. They play much like kids. Like kids at a playground or at a park, they'll actually have these interactions that are very quick that we can observe and quantify. They play tag, they wrestle. Their play is often likened to how dogs play or toddlers. And we can test whether our various genetic and environmental manipulations actually affect the quality of their playtime. And again, this is basic, but it's still really related to the human experience. And finally, some of my current work is looking at social anxiety. So mice are not naturally socially anxious. They're actually naturally social creatures, but we can actually induce social anxiety by what I like to call the bad time at a party. So you're a little mouse and you're going to a party, and you build up all your strength and you interact with this mouse and you go, hi, and you tell them a joke and they go, that's not funny. And you go, that's bad. I don't like that. I'm going to stay away from making these jokes. And after a couple tries of these mice making an effort to socialize and there being a negative reinforcer, in humans, it'd be, that joke's not funny, get away from me. In mouse, it's a little foot shock. They learn, we don't like social. We're going to avoid social in the future. And we have another group that gets to play with the other mouse at the party. And the other person says, that joke is great. We're best friends now. And they don't actually learn this social anxiety. And then when we take them away from the party and bring them to a new party with a new party mate, we can actually see, are they willing to engage again? Are they willing to continue going to parties? And are they willing to continue having fun? And what we find is that on the top, these are neurotypical mice. They actually, even after they learn this social anxiety, they let go of it. They're willing to go back and start re-engaging. And maybe it sucked the first couple of times, but they're willing to go to some more parties and try again. On the bottom, however, though, our genetic risk factor mice, they don't ever let go of the social anxiety. Like a lot of people on the autism spectrum, they just go, I don't want to do that ever again. I'll stay home next time. And again, different, but very similar. And what I'm going to leave you with is the tiny steps 5 through infinity, which are combining all of these different things, looking at different risk factors, looking at different kinds of social behavior to ultimately develop new drugs to target social anxiety and different kinds of social behavior that's relevant for autism and also the different kinds of social anxieties. Thank you.
Good afternoon, everyone. My name is Vivian Lau, and I represent the Ion Channel Drug Discovery Group based at the Brain and Mind Center. And so, oh, I forgot to tell you about the title of my talk. So today I'm going to talk a little bit about unraveling the mystery of untreatable epilepsy. So epilepsy is a brain disorder that causes recurring unprovoked seizures, and severity can range from 1 to 2 seizures per month to more than 2,000 in a day. And it affects 150,000 Australians, and that equates to 50 million people worldwide. And 1 in 26 people will develop epilepsy during their lifetime. Why should we care about epilepsy? Because it's the second most burdensome neurological condition after dementia, with an estimated global economic cost of about $180 billion per year. And most importantly, up to 40% of patients with epilepsies are resistant to current anti-seizure medication. And for this group of patients, they rely heavily on carers and often require 24-hour care. So therefore, effective treatment is an urgent unmet need. So we can, but then the necessary first step to find treatment options for these patients is understand the mechanisms behind drug resistance. So in 2022, our research identified a novel cause of treatment-resistant epilepsy. So today I will present our findings and outline the ongoing work aimed at finding treatment options for people with this form of epilepsy. So the textbook explanation for the cause of epilepsy is excessive electrical charge, and this is caused by hyperexcitation of the brain due to imbalance of the excitatory and inhibitory system. And this is a diagram showing the balance of inhibition and excitation of the brain. So we have GABA shown in orange, which is the major inhibitory neurotransmitter of the brain. And this is balanced by glutamate shown in blue, which is the major excitatory neurotransmitter of the brain. So in a happy, healthy brain, this balance between inhibition and excitation is intricately controlled. So when we have a little bit of a controlled excitation, we're feeling alert, we're going out and solving everyday problems. In contrast, when we have controlled inhibition, we get a little bit sleepy and we're ready to full rest. However, when this balance is disrupted by excessive excitation, and this could be due to reduction in GABA inhibition, epilepsy occurs. So GABA inhibition is mediated by a type of protein called the GABA-A receptors. And GABA-A receptors are ion channels. So ion channels is a type of protein that allow a flow of ions from outside of the cell to the inside of the cell. So excitatory ions are positively charged, whereas inhibitory ions are negatively charged. So chloride ions are negatively charged. So this is a diagram showing you the GABA-A receptor in blue sitting here. So it's sitting on the surface, the cell membrane of a neuron. And over here, so the blue blob, I don't know if you can see my arrows. So the blue blob is the GABA-A receptor sitting on the membrane of the neuron. And on the outside, we see GABA. Outside the cell, we see GABA and the chloride ions floating around. And what happens when two GABA molecule binds to the receptor, this triggers a response that opens the ion channel. And when the ion channel opens, negatively charged inhibitory chloride ions then is able to enter from the outside of the cell to the inside. And this negative charge, once it's inside the cell, inhibits neuronal activity. So mutations in the GABA-A receptors are found in patients with a diverse type of epilepsy. So they range from mild treatable fever-sensitive seizures to the severe drug-resistant epilepsy. And this is thought to be caused by the loss of GABA-A receptor function caused by these mutations. So less inhibition, more hyperexcitation. So our research question was at the time, if epilepsy is caused purely by the loss of inhibition, why do we see such a diverse range of epilepsy due to mutations in this inhibitory receptor? So to address this question, we study over 300 mutations found in patients, and we determine how each the mutation affect the function of the GABA-A receptors. So we use frog eggs to produce and study human GABA-A receptors. So this is the type of frog, this is the eggs this frog produced, and this is the type of frog that the eggs came from. It's called Xenopus lathis, also known as African clawed frog. So how we study these receptors in the frog egg is that we artificially engineer the mutations into the genetic material that makes up the GABA-A receptor. And this genetic material is then micro-injected into the frog egg, shown in the diagram. And then the frog egg is able then to use this genetic instructions to make the GABA receptor. And then that receptor is put onto the surface of the frog egg. And then once those mutant receptors are expressed on the frog egg, we employed this technique called 2-electrode voltage clamp, as you can see in the diagram on the right. The frog egg is clamped in between 2 electrodes. And this technique allows us to detect the amount of chloride ions flowing through the GABA receptors when we apply GABA. And on the left, you can see the experimental readout. So when we have increase of GABA concentration, we see the size of current amplitude that increases, and that indicates an increasing flow of inhibitory chloride into the cell. So when we're assessing GABA-A receptor mutations, we're evaluating this current change compared to normal GABA-A receptors found in healthy individuals. So in this work, after we studied over 300 mutations, we found 50% of the mutations actually cause the expected decrease in inhibition, so hyperexcitation. So that's what we expect, because epilepsy is a hyperexcitatory disease. However, to our surprise, we found the remaining 50% of the mutation cause increase inhibition, so hyper inhibition. So this is a paradigm changing discovery, and we published this paper in 2022. And so interestingly, these mutations that cause hyper inhibition experience more severe epilepsy and poorer clinical outcome. So these patients with the mutations have more severe epilepsy. They have early seizure onset. They have more drug resistance, and a significant percentage of these patients have what we call microcephaly, so a small brain. And showing this picture on the right, you can see this is an MRI of a patient with this hyperinhibitory mutations. You can see the brain is full and it's filling the skull. And this is the patient at 7 months. And when the patient is 7 years old, you can see the significant brain shrinkage over time. So it's filled with black space. So the brain has lost significant volume. So what is next for our research is that we want to understand why overinhibition also causes epilepsy and why does it result in more severe outcome. So existing epilepsy treatment focus on enhancing inhibitory pathway of the brain. But so therefore we need new class of drug for patients with overinhibition due to hyperactive GABA-A receptor. So to find treatment options for these people with hyperinhibition, we have 2 ongoing projects at the moment. So we're using our frog eggs as assays to screen for new drugs that could reverse hyperinhibition. And in another project, we're currently developing brain organoids to study those mutations. And brain organoids are essentially miniaturized brain grown in a dish from stem cells. So using these organoids will enable us to investigate the impact of those mutations in a more complex system, so more complex than frog eggs, in a more controlled environment. And hopefully this will provide mechanistic insight into how hyperinhibition can also cause epilepsy. So that. Yes, thank you.
Good afternoon, everyone. I'm Nathan, and I'm thrilled to be here today to talk to you about how to save a memory. And I'll begin with an introduction. So this is Tim, and Tim likes doing many things, from playing guitar, playing games, and playing sports. I'm not sure if this is playing. It is playing? Oh, okay. Well, Tim's buffering indeed. Okay, this might be better. Tim likes playing guitar. He likes playing games, and he likes playing sports. And Tim also is really, really into books and spends a lot of time reading and learning new information. But there comes a point in every day where we feel tired, and at some point Tim has to go to bed. But Tim comes back the next day and he's back at doing all his favorite activities all over again. But how is Tim able to pick up where he left off and continue to progress and improve and generate new ideas and not sort of forget what he's done and not improve? Well, if you haven't guessed by now, he does that through memory. And how are memories formed and saved in living, breathing organisms like you and me? Well, that's done through a process of transforming experiences into concepts. We'll start with an example. So what are these images of? Are they all the same tree? So how are we able to learn, firstly, what a tree is and what are the different types of trees and what makes them different? Well, our memories work in a way that sort of blurs the concept, blurs the experience and transforms it into sort of the stereotypical concept that we're able to use in future. So we go from a really high definition experience with lots of full detail, and over time we generalize it into a concept so that we're able to explain or identify a tree based on some features like a trunk that goes into the ground and a big bushy green thing at the top. and the differences between different types of trees. Maybe there's like red fruit growing from it, or the patterns or the shapes of the leaves, or just the shape of the tree in general. And as a neuroscientist, we're interested in understanding how our brains transform these experiences into concepts. So here I'll introduce you to a little brain region called the hippocampus. And the hippocampus is really active and involved when we're dealing with novel situations and experiences. And it's really good at that because it has these neurons, like the ones you're creating, that are really fast acting and quick firing and able to adapt to novel new situations. And then we have this sort of outer layer of the brain, which takes up about 80% of the mass of the brain. It's called the neocortex. And that's really got a lot of generalized functions that it does. So there's lots of regions in there that do specific things, but it's a very generalized brain area. And it's more involved in sort of us understanding concepts and being able to adapt and generalize things. And how do we get from that experience to the concept? Well, you can think of the hippocampus like Geppetto and the neocortex a bit like Pinocchio. And when we're early on in an experience, these quick firing hippocampal neurons are sort of pulling the strings on the cortex and telling it what's happening and how things are connected. And over time, the hippocampus gets less involved and sort of the memory or the concept comes to life in the cortex and is able to be trained to understand. And does this through replaying those experiences over and over again after we've experienced them. But when does this replay happen? Well, for that, we'll come back to revisit Tim. So let me just make sure this is buffering. Yes, it works. So at the end of the day, when he gets tired, Tim goes to bed and he drifts off to sleep. And as he sleeps, he's not only resting to recover his energy for the next day, he may dream of fantastic things that are happening to him, but there are also things going on that he's unaware of. And as he sleeps, his brain is actively working away. And this activity that's going on involves communications between his neurons that are working to transfer information around in his brain. And if we were to put a little sensor on his head and measure this activity inside, this is what the brain activity would look like while he's deep asleep. It's quite slow, big amplitude. And indeed, that's quite different from when Tim's awake, where his brain activity looks like that. And so by measuring brain activity, we're able to really determine what sort of communication and activity is going on in the brain. And indeed, using brain activity is one of the best ways currently to measure sleep objectively. And as we sleep, we go through different stages of sleep, which are represented by different patterns of activity of our brain signals. And there's two main classes. There's rapid eye movement sleep, which you've probably heard of, and that's because it's defined by eye movements that are occurring, but also this fast frequency brain activity that looks a bit like when we're awake. And that's where you have these vivid like dreams. But there's also this other stage of sleep called non-rapid eye movement sleep, which have these other distinct waveforms going on. And inside these sleep stages, there are these very distinct sort of events that occur. They're called brain oscillations, such as sleep spindles, which are termed because they represent reflect a spindle of yarn. And we also have these brain waves termed slow oscillations. They're big, nice, slow waves. And what do these brain activity patterns represent, and why are they useful or informative? Well, because they are generated in different parts of the brain. So these nice, big, slow oscillations are occurring in the outer neocortex. And these sleep spindles are occurring in another part of the brain called the thalamus, but they're very tightly linked to this hippocampus that was involved when we experience things. And what's interesting is these brain activity patterns are timed to each other, and they sort of indicate the right time for each other to occur, such that these nice big slow oscillations time these faster or medium frequency spindles And then if you zoom in on the spindle inside, you have the hippocampal really fast activity going on during that. And this is this timing of this communication between the different brain regions while we sleep resembles that sort of puppetry going on between the hippocampus and the cortex. And what I study is what happens when we get older. So as we get older, our brain activity changes. And what's one of the biggest changes that occur during our sleep as we get older is that our brainwave amplitude decreases. So when we're younger, we have these nice, big, slow amplitude waves. And as we get older, the amplitude decreases. Why is that? Well, it's nicely linked or correlated with the presence or the occurrence of these things called amyloid plaques, which occur attached to neurons, destroy their integrity, their flexibility, and make them less flexible to activate. And this happens everyone gets older, we all get these little plaques, but there's quite an extensive buildup of them in disorders like dementia or Alzheimer's disease. But another thing that really happens in our brains as we get older, it's very important for this cross-communication and memory saving, is timing. And to represent that, I'll show you this short video. So this is a video of surfers out in the ocean. And nowadays, to help them catch a wave at the right time, there's this little platform and they can jump off and catch the wave perfectly at the right time and enjoy their surf and have a great day. So if we take that metaphor as our brainwave activity, and we think of the little surfer as the sleep spindle, which is related to the thalamus and hippocampus, and we think of the wave itself as the slow oscillation of the outer part of the brain, the neocortex. Well, in younger adults, the little sleep spindles are catching the wave right at the perfect time so that they can transmit that information across from different brain region to brain region and communicates that information effectively. But as we get older, the spindles are kind of catching that wave a little bit too late. They're not able to catch and surf, and so they're missing out on transferring that information, and all hell breaks loose. Okay, the information is not being transformed effectively. And this is really well studied, and that's what I study, is this coupling of brain rhythms and how it changes across the lifespan. So as we mature from children to adolescence, this timing becomes earlier so that the little spindle is able to catch the wave at the right time, just before the peak of the wave. And as we get older, the timing gets later and later. And as people exhibit cognitive decline, hippocampal damage, or neurodegeneration, it becomes later and later and later. And that disrupts that effective communication that's really so important to save memories as we sleep. And that has been shown to be related, the timing of these oscillations have been shown to be related to memory performance in many studies. So what can we do? How do we improve brain rhythm? Well, there's thankfully, there are some good researchers out there trying to do this, to improve this. One technique people have come up with is called acoustic stimulation. And that, how that works is we wait for people to fall asleep, we measure their brain activity, and we wait for the time that we expect one of these slow waves to occur, and we just play a tone. And if you play a little sound, like a little beep, people don't wake up, they don't hear it, but their brains recognize it, and it boosts another few slow waves to occur afterwards. So we're able to kind of trick the brain into having these nice slow oscillations. And that works really nicely, but there's been some recent evidence come out that shows while it works for enhancing brain rhythms, it doesn't actually impact our memory function at all. So unfortunately, we have to think of something else. Another really novel idea is actually rocking people to sleep. And this one, you're very lucky tonight because this research was done by my wife and she's in the audience here. And basically how this works is you put someone into bed and you laterally rock the bed very slowly at a specific rhythm. And what they were able to show was that it improved sleep quality, but it also timed, compared to when the bed was stationary, it timed the onset of these slow oscillations to occur at specific times when the bed was rocking. And really nicely, It also improved memory performance overnight. Now, this sounds great. It was only done in healthy young people, and research is ongoing. We're trying to do this, or people are trying to do this in older adults to see if this actually can improve our memory as we get older. So stay tuned. Maybe in the future, everyone will be rocking themselves to sleep. So to sum up, memory involves transforming experiences into concepts. The brain areas involved in experiences talk to each other to learn and train concepts. And I hope you can take away that timing is everything. And as we get older, the timing of this brain conversation is delayed, which then impacts our communication. Thank you.
Okay. Thank you very much for having me here today. I'm very delighted to be part of this wonderful event. So today I want to take you on my journey as a scientist. And it all began in high school when I fell in love with biology. I was always curious about how living things work. So naturally, I pursued a science degree at university. During my studies, I became captivated by the brain. And this fascination led me to pursue an honors degree in psychology. And I had the chance to explore how oxytocin, often called the love hormone, how it affects social behavior, and not in humans, but in rats. So yes, those little guys in the lab taught me a lot about how we connect with each other. Now, my passion for understanding the brain didn't stop there. I went in to do a master's in clinical neuropsychology, where I dove deeper into the mind, studying how it influences our thinking, our behavior, and our cognition. And this experience was a real light bulb moment for me. It made me realize I wanted to pursue research looking at finding novel treatments that will improve our well-being. So this led me to pursue a PhD looking at a new topic that's becoming very popular and more relevant today is how medicinal cannabis can help treat chronic illness with a specific focus on chronic pain and sleep disorders. And this experience got me thinking, how do these conditions change the way our brains work? And how do they affect our memory and thinking? So all of these experiences have brought me to where I am today. So I'm, I'm a senior researcher at the Healthy Brain Aging program within the University of Sydney, where I'm exploring the effects of chronic pain and sleep disturbance as emerging risk factors for cognitive decline and also dementia. So what is dementia? So it's a progressive brain disorder that affects your memory, your behavior, and everyday life. It is the second leading cause of death in Australia, with Alzheimer's disease being the most common type of dementia. Now, it's caused by a mix of factors, including our genes, our biology, and the environment that we live in. And these factors combine in complex ways to affect our brain health. Now, one of the most surprising things about dementia is that the brain can start showing subtle signs of damage up to 20 years before the condition is diagnosed. So that gives us a significant window of opportunity to potentially change the course of the disease. So what can we do? Now, you'll be surprised to hear that about 45% of dementia cases can be potentially prevented by addressing 14 key risk factors. And this is incredible because it means that there are things that we can do today to reduce our risk of developing dementia down the track. Now, some of these risk factors are well known, like managing high cholesterol, staying educated and socially connected, and getting enough exercise. But there are also some lesser known ones, such as treating vision and hearing loss and reducing our exposure to air pollution. So overall, this shows an enormous potential for prevention. And so as part of my research, I'm focusing on two emerging risk factors that we're just beginning to understand, and that is sleep disturbance and chronic pain. Now, chronic pain and sleep disturbance place a huge burden on the brain and cognition. Both impair our thinking skills, our concentration, and also our mood. And they also exacerbate each other, so they make each other worse. When someone is in pain, it's harder to sleep. And when someone isn't sleeping very well, pain often feels more intense. And they also contribute to inflammation, which is a key factor in the development of Alzheimer's disease, suggesting a shared biological mechanism. So in my research, I'm focusing on how these two conditions influence each other and influence cognition and the risk of developing dementia. But let's rewind a bit. What happens in the brain as we age and in dementia? This is a close-up image of neurons in the brain's hippocampus. So this is the seahorse-shaped region located deep inside the brain, sitting on either side of the brain, and it's an important region for memory and learning. Now, the blue color indicates the main parts of the cell. So this is called the cell bodies. And the red lines show, or the red color shows the branching structures of the neurons that help send signals in the brain from neuron to neuron. So these are axons and dendrites. Now, the bright green spots represent the active centers inside the cells, where a lot of important brain activity occurs. Now you've seen what a neuron looks like in the brain. Let's see what happens to these neurons in dementia. As we age, two proteins called beta amyloid and tau start to build up in our brains. Beta amyloid forms clumps outside the brain cells, disrupting communication between neurons, while tau forms these tangles inside the brain cell, causing damage to the structure of the neuron. And these changes are key factors in the development of Alzheimer's disease. And you'd be surprised to hear that we all start to accumulate these proteins as early as age 20. However, only those with certain risk factors will go on to develop dementia. Now, neurons aren't the only cells that are affected. The brain's immune cells, are called astrocytes and microglia, also play a critical role. So these are the brain's cleanup crew. So they're clearing out waste, they're trimming unnecessary connections between neurons. However, in dementia, they get overwhelmed by the beta amyloid and tau accumulation and become overactive, releasing harmful substances that can damage neurons and start removing healthy connections. And this is called inflammation in the brain, or neuroinflammation. So this is very important to track progression of the disease and also monitor how things are happening in the brain. But how can we visualize this in humans? So our research uses a special brain scan called a PET scan. And similar to other scans, people would lie in the scanner and it takes a beautiful image of their brain. And what this allows us to do is to visualize the buildup of these abnormal proteins and also inflammation in the brain using different types of tracers. So this helps us to not only diagnose dementia more accurately, we can also intervene early by identifying individuals who are at risk of dementia. So in the middle panel, panel B, we've got a group called mild cognitive impairment. And this group has cognitive problems that are identified on cognitive testing. And 45% of these individuals will progress to dementia within 5 years. So being able to identify this group allows us to intervene early and enroll them into research in order to change the progression of that disease. And so by combining this data with essential medical, cognitive, and other biomarker data that we collect in the clinic, we can better understand how certain risk factors such as sleep and chronic pain influences these brain changes. And the ultimate goal is to discover new treatments. So the final part of my research focuses on adapting an evidence-based digital sleep intervention. So this is an intervention that has very good quality evidence supporting its effects in improving sleep. And we're going to adapt it for a chronic pain population. So we want to determine whether this intervention can improve symptoms of pain and sleep disturbance in older adults who have cognitive impairment. And down the track, we also want to see whether this intervention can slow down cognitive decline. And so the ultimate goal would be to prevent or delay dementia. So thank you so much for listening.
Okay, so now we've reached the Q&A portion of the talk. So I'd like to invite all the speakers to come up. And if anybody has any questions, just, in person or online, put your hand up if you're in here. And then online, if you could just submit your questions via the Q&A, and I'll get out of the way. Yeah, so I've got a question over here. I'll just bring the mic around to you guys. If you could just speak into the mic, and then everyone online will be able to hear you as well.
Hi, that research about dementia was really informative. I have relatives several relatives with dementia. Is the current state of science that dementia can be prevented?
It's a great question. And what we found recently, there's been a recent commission into this called the Lancet Commission that looks at these modifiable risk factors for dementia. And what they found was that 45% of dementia cases can be potentially preventable if we address those risk factors. So it does highlight that we could potentially halve the number of cases of dementia if we can intervene early and look after ourselves. So a lot of those risk factors are very, as I mentioned, very common things that we would do, such as regular exercise, managing our weight, lowering our cholesterol. But there's other things as well that could be addressed, and so it's definitely an important initiative to target those.
Really, really helpful. Thanks.
Thanks for your question.
Thank you. Does anybody else have any questions? Yep.
Thanks. I had a similar question. It was great to see all the links between your talks. I thought I'd ask this question before I forgot. I'm thinking about getting a houseboat now after seeing the rocking bed. Have I got the right idea? Would that work?
It's a great question. It's one that always comes up. But there is some truth behind it, because often we find that we can fall asleep easily in a car or on a train or in a hammock. But for the benefit of memory, the thinking behind it is that the rocking has to be very slow and at a specific frequency and very, very uniform. Otherwise, you might actually have the opposite effect and disrupt your sleep. So a houseboat, yeah, it might help you fall asleep quicker, but the jury's out whether or not it actually will help with these memory processes that we're aware of.
Yep, got one over here.
Thank you. I'm not sure if you can answer this, but I was thinking about teenagers and their reliance on devices when they're going to bed and their patterns in going to sleep, and what are they dreaming, and is that going to affect them? Are they going to not be able to surf those waves as well younger? I don't know whether the research is going down that route yet, but we older people would like any ammunition at all to get our children and grandchildren off these devices, sort of at bedtime.
Yeah, I think I can start with that one. What I spoke to about sleep is one function of sleep, and that's all about the timing and the memory consolidation, and having good quality sleep is really important for that. But we know that sleep's also important for a lot of other things. And so indeed, disturbing your sleep with screens before bed can impact the timing of sleep. And if you're not having sleep at the right time, that can also have impacts for other things as well. And sleep, yeah, it's not just important for our memory, but it's also important for preparing our brains for optimal learning the next day. So sort of resetting. And there's also some evidence that it's really important for cleaning out sort of these toxins, these things we talked about, the amyloid plaques and the tau tangles. There's some emerging evidence that sleep's actually important in cleaning those out as well. So it has a lot of functions, and it's not just for our brain as well, it's our body too. It's a very complex behavior and function. But indeed, I think it's very important that we monitor how well people are sleeping and make sure that they're getting the optimal sleep without going too far over and making people anxious about their sleep as well. So we hear a lot about you have to sleep well, you have to sleep well. But some people then might start to think, oh, I'm not sleeping well, is that an issue? And that can cause more anxiety and more stress. So it's about kind of understanding that how your sleep should be roughly, but that everybody's different and that we all have different needs as well. So yeah. I don't know if anyone wanted to add anything else.
Okay.
Yeah.
I'm particularly interested in Parkinson's and brain disorders because, I mean, my husband has had Parkinson's for 17 years, and recently he's had horrendous hallucinations, and it turned out it was being caused by an imbalance in his medication. So, and also I'm interested in Parkinson's and dementia, etc., because I believe they're sort of related, or is it related because of the medication?
I'm not entirely sure any of us are qualified to answer that. The brain is complicated and we're only 4 people.
Yeah.
Okay.
Is there any relation between meditation and keeping the memory going, or is it similar to sleep, or has there been any studies on that, if you know of?
Not that I'm aware of, but I think there are some studies showing that meditation can help with sleep. So therefore it might help sleep quality, but I don't think there's any evidence on meditation for memory, unless anyone else knows of any.
I think something like meditation and mindfulness practice would target some of the risk factors that are linked to dementia. So things such as sleep disturbance, that's an emerging risk factor, but also depression and your general mental health. So all of it interconnects. And so something like that, I think, would be very important for your overall well-being. And that will also tick off some of those risk factors that are the 14 on that list. So although there is some emerging studies looking at mindfulness intervention as a treatment for sleep, so insomnia, and that's definitely shown to be quite effective. Whether it's then that leap between, has it then changed the disease trajectory in people with maybe mild cognitive impairment, we don't yet have that data. Okay.
Hello, thank you for your presentation. Since you tricked us all here on the premise of knitting, is there any link between knitting and positive effects on the brain?
Well, I think that links really nicely with the previous question, really just doing something mindful where you're coming together as well and socially connecting, engaging your mind and trying something new, I think is very important for the brain, and the brain loves that. So the more you do that, the healthier your brain is, the more connected your neurons are, the more they fire and wire. So certainly, definitely a great activity for something like that.
I will say that there are some studies in kids, and especially kids with ADHD and autism, who typically have a hard time sitting down and doing something. Giving them a focused physical, tactile activity actually improves other areas of their cognitive and executive functioning. So I think they've done some studies where they have got kids doing puzzles or something while listening to lessons or something, and those kids ends up actually absorbing more than if they were just told, you're actually going to go to this lesson and just listen. So I think there is something about the duality of doing something tactile with our hands, but having our other senses available to us, especially when it's something simple, repetitive, kind of almost tactile pleasing as well. So I think it's not just about aging too, it's like during development, it can also help too.
We do have some questions from online, so we'll just get Nima at the back here to read them out to us.
Robert asks, should people over, say, 50 do regular brain scans to detect potential dementia? And what would be, if so, what would be the appropriate frequency?
Okay. So generally speaking, if someone is noticing changes in their memory and thinking skills, that should be the driver for a consultation with their GP. And so it's not necessary to undergo these scans, because we all develop and accumulate those toxic proteins as we age, and that's a normal part of life. It's only when you have those certain risk factors, and again, that's a very complex interaction that can then lead to dementia. So if someone is experiencing changes in their cognition, their thinking skills, and they're worried about it, or someone else in their family has noticed, that should be the driver for seeking help. Certainly not to undergo unnecessary brain scans.
Thank you. Thank you. And Lynn asks, what types of epilepsy are best assisted with cannabis? If that's relevant, otherwise we can go to the next one.
I can answer that. So doing a PhD in cannabis allows me to, well, and Vivian, you would know as well, the best evidence we have for using cannabis for epilepsy is high doses of CBD or cannabidiol, which is the non-intoxicating component of the cannabis plant. And we actually have a registered product in Australia called Epidiolex, which is a highly purified cannabidiol oil that is prescribed to children with severe epilepsies. So CBD has the best, highest quality evidence in reducing seizures.
Thank you. And Anonymous asks, how do we deep sleep? Does dreaming mean we sleep deeply?
Well, good question. Deep sleep, it can be defined by how responsive you are to being woken up. And when you're having these nice rhythmic brain waves, then you're in what's considered the deepest sleep. The REM sleep is also sort of a moderately deep sleep, but your brain activities, even though it's more like wakefulness, your body is fully paralyzed. So your brain's going rapid fire, having these really vivid dreams, really active brain states, but your body is completely still. So it depends on your definition of deep, but yeah, in terms of responsiveness, the slower brain waves means you're more deeply asleep. And the REM is also a deep sleep state with a completely different function that is crucially understudied, honestly.
Thank you. And Anonymous also asks, for young people in high schools with an interest in the area of biology or neuroscience, is there a capacity, work experience in any of the labs where the research comes from? Thank you.
Always. There is always a need for young people in labs and in science. A recommendation is if there's an area of research, or even if you're near a university, email people. That's how I got into research. I emailed 65 professors at the hospital where my mom worked at, asking, I am an undergraduate student. I have no skills. Will you take me for a few weeks? And only one responded. But he gave me a chance, and it totally changed my mind. I was going to become a physician. And I said, I actually think research is my calling instead. So do not be afraid to send heaps of emails. Many of them will get ignored, but some of them will not. And there's tons of people who will definitely give opportunities.
Thank you. And just one last question from Alibu 333. You talked about early intervention for looking after our memory. Is there a link for early detection for neurodiversity and treating it early?
Oh, big question. I mean, neurodiversity is also a big word. Diversity is because it's diverse. A lot of, especially for autism related diagnoses, it's a lot of like asking parents to look for earlier milestones. So we talk about those social aspects, you know, do your children make eye contact with you? Do they respond to their name? It's actually those earlier milestones that can actually be bigger warning signs. So my PhD was actually in motor disability, and one of the earliest and most accurate markers of whether a child will be diagnosed with autism is actually whether they'll be able to pick up their neck at the appropriate developmental milestone age, and whether they're going to learn how to crawl and walk. A lot of parents go into doctors and say, my baby's not crawling or not picking up their head or not orienting towards me. And now physicians are actually advised to take those really seriously and start monitoring those kids, because, you know, 4 years old is, it's very young, but it's also couple years of missed opportunity to give occupational therapy. There's heaps of studies showing that those kids who show those early motor disabilities, if you give them some occupational and physical therapy as babies and toddlers, they actually have a less severe disorder as they grow up. So those earlier time points and capturing disorders early can actually make a bigger difference. Now, that's one kind of, you know, neurodiversity. We have kids who will never have any motor disabilities and will have only social struggles or anxiety struggles later on. You know, it can't always help everyone. Same thing with a lot of people develop or to see the signs of ADHD much later than, you know, toddlerhood or very early childhood. So there's a lot of like parents actually being really honest with what's happening with their kids and being honest with their GPs, and their GPs also being open to that feedback, because they're often parents know their kids best and they know what's best for them too.
Okay, thank you. We'll take one more question. Oh. Sorry, did you have a, did you have a question? Okay, cool, just checking.
You were saying that hearing loss and vision impairment was a warning sign, or what if people are born deaf or blind? Is that an issue in
That's-
regards to dementia?
A great question. And since the release of this new guidance describing all the different risk factors, I had the same question and I haven't yet looked into it. So I think that's a really good point. And a lot of people also experience hearing loss and changes in their vision and don't go on to develop dementia. So there's many, we don't actually understand the mechanism that drives that. It's still unclear. So unfortunately, I can't answer that question, but it's a good one.
Okay, we are at time today. Sorry, I'll find a slot in somewhere up here. Yes, we are at time today, but thank you all for joining us for the talk today, and thank you to our speakers as well. Please stick around for the next half hour as we'll continue to knit neurons. We'll also have the speakers sticking around, so if you'd like to have a chat or if you have any questions for them, just stick back for, we'll be here for another half hour. We have 2 more talks coming up in October for Mental Health Month. So if you enjoyed this talk, we'd love to see you there as well. Okay, so thank you. Have a lovely evening and we'll see you next time. And thank you to our speakers.